Now, what is next? Of course, the next part will be to how to plot the points correctly. Now, this is a no-brainer. As long as you have the table next to your graph or near your graph, you can actually plot the points quite easily, step by step. So remember to use a pencil to plot carefully. Make sure that all the data points are in, all the pHs are in this case. Of course, very seldom we'll have 14 points. Most of the time for an exam, you get about 5. And so it'll be fairly easy. And after that, just join up the line. Well, I know it looks a bit ugly. Well, that's the best I can do using a computer. But if you use your flexi curve or even your French curves, and you do a decent drawing of the joining of the dots, like I used to say, it should be fine. And remember not to use a ruler to join up dot by dot. Make sure that the curve is fairly decent. Make sure you use a single pencil line. I think as long as you do all these, it should be fine. So a few things to remember, of course. Don't forget to label the axis with units, right? So in this case, remember that the x-axis is the pH, right, and the y-axis is the time taken. So remember to do these things because it will cost you marks. So what do markers actually look out for? Now you can remember this simple acronym called PALS, right? The markers usually look out for these few things. Number one, they look out for points, whether you plot the points correctly, whether you cross it and all the points that are given in the table are plotted. Second, look for the axes, whether the axes are labeled, okay, whether they are marked, whether they start at the 0. Remember, you don't have to start at 0. You can start at any value you want to. But please don't use the squiggly line that you use for mathematical tables, because for biological graphs you actually require to you can use the chart, or use the graph, sorry, to predict values, right? So you don't use that line. For the line is how you join the points together, whether it's a one continuous line, whether you do it rather smoothly, okay, and you either use a pencil, and of course last but not least is the scale, whether the graph occupies more than 50-75% of the graph paper. So this is the end of the first part of the lesson, which is on graphing, and the second part is actually about analyzing graphs. Now, for analyzing graphs, the first thing that you really need to do is to understand what the graph is about. Most of the time for biology, the graphs could be enzyme reactions, simple pH versus activity or time taken, or temperature versus time taken. The other one is, which you most commonly encounter, is actually osmosis and diffusion types of questions, where you put a certain size of potato strips or something, and the potato strips change in size according to the concentration or the water potential of the solution. And of course, another one that we did during SPA was actually standardization curves, whereby you're given different concentrations, and you see concentrations of, for example, glucose, and you see whether they break down, because glucose is reducing sugar, whether it breaks down the potassium permanganate, or how long it takes for it to make it clear. And then using that, you can predict values. And of course, the next thing that you really need to do is find out where are the maximum and minimum points, which should be relatively simple if the curve only has one. So next thing, once you find out the maximum minimum points, it's good to split the curve in half, right, if possible. And then after that, a simple description and explanation of the curve from start to the maximum minimum point, and then from the minimum point or maximum point to the end of the graph. So split the curve in half and describe these two halves, right? From increase from the start to the middle, and then from the middle to the end. So I'm gonna do. Other points of interest, of course, is that there are two curves on the graph that require some. If there are two curves on the graph that require some comparison, you need to do some work and compare. And always remember to describe and explain. Explain why. That's why it's important to understand what the graph is about. So I'm going to do a simple worked example, basically using two curves, something similar to what you've done in the Essay 1 for the Sec 4s. So first and foremost, you split the main curve in half, as you can see the blue line, the blue curve. I mean, you can kind of draw an imaginary line between the two. So seems to be this minimum point at pH of 7. So after that, you describe the curve from the start to that point. And as you know, the curve is decreasing, right? So you know that the time taken is decreasing from pH 1 to 7. Now please remember, as the time taken increases, right, the rate decreases. So they are inversely proportional to one another. So pH 1 to 7, time taken is decreasing, which means the rate is increasing. And then from pH 7 to 14, the time taken is increasing again, and that means the rate is decreasing. There is another curve on the graph paper. In this case, it's the 60 degrees one, where you can't find really a maximum or minimum point. So what you could do is you can describe it by saying the time taken is fluctuating between about 100 seconds to 120 seconds with no clear maximum minimum point. So this gives a rough description of what the green curve looks like. So where possible, describe fully the curve for the blue one, especially when you can see quite a prominent minimum point. So split in half based on that minimum point and describe the two ends. But for the green curve, if you can't see really what it looks like, then just do a rough description whereby it's fluctuating or it's increasing and then decreasing, and explain that there's no clear maximum minimum point if there are more than one. Okay, so remember to explain, right? So it's important to show, not just to describe, but to explain why it's like that. Now, for you to explain properly, you definitely need to understand what this curve is about. And as you can see, it's about pH, changing pH and measuring the rate of reaction. So you should know that this is an enzyme reaction, right? And you know, because it's an enzyme reaction, is affected by temperature and pH. So remember to use words like optimal pH, optimal temperature, enzyme is denatured or inactive, or it's the best temperature for it to work. So remember to always use words that are linked to such an example. So it could be many examples. It could be osmosis diffusion type. So remember to use words like water potential, or water going in by osmosis or leaving by osmosis, or because the water potential outside is more or less compared to the inside of the cell. So always use words that are linked to the topic, to the main topic, and use those to explain the graph. Try not to use your own words because they may not get you marks. So that's about it. It's not too difficult. I hope it helped, and all the best for exams. Thank you.